The neuroscience of social media. How digital platforms reshape our brains. In a time when our digital lives and real-world experiences are closely connected, it's more important than ever to understand how our brain works when we are online. Today we take a closer look at neurons, synapses and brain circuits to see how social media is changing the way our brains function. From the moment you get a notification to the urge to check your feed, a series of brain processes is happening. We'll explore how social media platforms are designed to tap into our natural reward system, affecting our focus and emotional control. But it's not just about quick bursts of happiness. We'll also look into how long-term social media use might be causing changes in important areas of the brain, possibly rewiring the way it works. Can these digital interactions actually change the structure of our brains? And what does that mean for our thinking, emotions and social behaviors in the long run? Join us as we explore how the brain and technology interact, revealing the hidden brain process behind our online experiences. By the end of the video, you will understand how social media impacts your brain at different levels and what it means for the future of human thinking in the digital world.
The neurobiology of reward, dopamine and beyond.
To truly understand how social media affects our brains, we need to start at a molecular level, focusing on the intricate workings of our reward system. While dopamine often takes center stage in discussions about digital addiction, the reality is far more complex, involving multiple neurotransmitter systems and neural circuits. Let's begin with the mesocorticolimbic dopamine system, a key player in reward processing. This system originates in the ventral tegmental area, short VTA, of the midbrain and sends signals to several regions, including the nucleus accumbens (NAC) and the ventral striatum, the prefrontal cortex (PFC) and the amygdala. When we experience something rewarding, like a social media notification or like, neurons in the VTA release dopamine. This dopamine binds to receptors on nearby neurons, setting off a chain reaction that changes how our brain cells behave and express certain genes. However, dopamine doesn't act alone. Glutamate, an important neurotransmitter, helps control dopamine release and shapes how we respond to rewards. The interaction between dopamine and glutamate in the NAC is crucial for reinforcing behaviors that feel rewarding. Recent neuroimaging studies have provided fascinating insights into how social media activates this reward system. For instance, a 2019 study using positron emission tomography found that receiving likes on social media was associated with increased dopamine receptor occupancy in the ventral striatum, similar to what's observed with addictive substances. Moreover, though, functional magnetic resonance imaging (fMRI) studies have shown that social media use activates the same brain regions involved in processing other types of rewards, such as food or money. This includes increased activity in the NAC, orbifrontal cortex, and anterior cingulate cortex, all key components of the brain's reward circuitry. But the story doesn't end with simple activation. Prolonged and excessive social media use can lead to adaptation in these neural circuits. For example, a constant overstimulation of the dopamine system can lead to receptor downregulation, potentially making it less responsive to natural rewards and driving compulsive behavior to achieve the same level of stimulation. Furthermore, a recurring and unpredictable nature of social media rewards, you never know when you get a like or a comment, creates a pattern of reinforcement similar to that what we see in gambling. This reward schedule is particularly effective at maintaining behavior and can lead to the development of habitual checking and scrolling. It's important to note that while the dopaminergic system plays a central role, other neurotransmitter systems are also involved. For instance, the serotonergic system, which is implicated in mood regulation, is also affected by social media use. Some studies suggest that excessive social media use may lead to alterations in the serotonin signaling, potentially contributing to mood disorders.
Neuroplasticity and structural brain changes.
Now that we have explored the acute effects of social media on the neurotransmitter systems, let's dive into the long-term structural changes that may happen in the brain with prolonged use. This brings us to the concept of the neuroplasticity, the brain's remarkable ability to recognize itself by forming new neural connections throughout life. Neuroplasticity occurs through various mechanisms. The first one could be a synaptic plasticity. These are changes in the strength of synaptic connections between neurons. The second one could be the neurogenesis, the formation of new neurons, particularly in the hippocampus. The other two important mechanisms are the axonal sprouting, the growth of new axonal branches to form new synaptic connections, and the dendritic remodeling, changes in the structure and number of dendritic spines. Recent neuroimaging studies have shown evidence that excessive social media use may indeed lead to structural changes in key brain regions. For instance, a 2018 study used voxel-based morphometry found that individuals with internet gaming disorder, which shares many features with social media addiction, showed reduced gray matter volume in the anterior cingulate, orbifrontal cortex, and prefrontal cortex, regions crucial for executive function and impulse control. Another study using diffusion tensor imaging, short DTI, revealed alternations in white matter integrity in the corpus callosum of individuals with high social media use, suggesting potential changes in the interhemispheric connectivity. These structural changes may have significant functional implications. For example, reduced gray matter volume in prefrontal regions could potentially impair executive functions such as decision-making, impulse control, and emotional regulation. Similarly, alterations in white matter connectivity could affect the efficiency of information transfer between brain regions, potentially even impacting cognitive processes that rely on distributed neural networks. Here, it's also important to note that the relationship between social media use and brain structure is likely bidirectional. While excessive use may lead to structural changes, pre-existing structural differences may also predispose individuals to problematic social media use. Moreover, not all neuroplastic changes associated with social media are necessarily negative. Some studies suggest that certain forms of online interaction, such as engaging with educational content or participating in cognitively demanding online tasks, may actually promote positive neuroplasticity, enhancing cognitive functions and potentially increasing gray matter volume in regions associated with learning and memory.
Attention network and cognitive control. One of the most profound ways social media affects our brain is through its impact on attention and cognitive control. To understand this, we need to examine the neural circuitry underlying attention and how it's challenged by the constant stream of information and notifications provided by social media platforms. Attention is not a unitary process, but rather involves three distinct neural networks. The alerting network, which maintains a state of vigilance and readiness to respond. The orienting network, which selects specific information from sensory input. The executive control network, which resolves conflict among responses and regulates thoughts and feelings. Social media platforms are designed to constantly engage these attention networks. Push notifications activate the alerting network, novel content engages the orienting network, and the need to switch between different types of content or decide whether to respond to a message activates the executive control network. However, the rapid and continuous engagement of these networks can lead to cognitive overload and fatigue. Neuroimaging studies have shown that multitasking, which is common during social media use, is associated with reduced activity in the prefrontal cortex and increased activity in the striatum, reflecting a shift from a goal directed to more habitual behavior. Furthermore, the constant switching of attention required by social media may be altering our capacity of sustained focus. EEG studies have found that heavy social media users show altered pattern of brainwave activity, particularly in the alpha band, which is associated with attention and inhibitory control. One particularly concerning finding is the potential impact on the default mode network, a set of interconnected brain regions that are active when we are not focused on external environment. The DMN is crucial for processes like self-reflection, autobiographical memory, and creative thinking. Excessive social media use may be disrupting the normal functioning of the DMN by constantly pulling other attention outward. This could have profound implications for our ability to engage in the deep thinking, self-reflection, and creative problem solving. Moreover, the brain cognitive control network, which includes regions like a dorsolateral prefrontal cortex and anterior cingulate cortex, may be particularly affected. These regions are crucial for a top-down control of attention, decision-making, and impulse regulation. Neuroimaging studies have found altered activation patterns in these regions in individuals with problematic social media use, potentially reflecting difficulties in exerting cognitive control over their online behavior.
Emotional processing and social cognition. Social media doesn't just affect our attention and reward systems, it also has profound implications for how we process emotions and engage in social cognition. Let's explore the neural underpinnings of these processes and how they are influenced by our digital interactions. Emotional processing involves a complex network of brain regions, including the amygdala, insula, anterior cingulate cortex, short ACC, and prefrontal cortex, short PFC. The amygdala, in particular, plays a crucial role in detecting emotionally salient stimuli and initiating appropriate responses. Neuroimaging studies have shown that receiving positive feedback on social media, such as likes or supportive comments, activates the amygdala and ventral striatum, similar to the way we process social approval in face-to-face interactions. However, the intensity and frequency of these digital social cues may be leading to a kind of emotional inflation, potential desensitizing us to real-world social feedback. Moreover, the constant exposure to carefully curated content on social media platforms can trigger social comparison process, activating regions involved in self-referential thinking such as the medial prefrontal cortex, MPFC for short, and posterior cingulate cortex, PCC for short. The heightened self-focus combined with the often unrealistic standards presented on social media can lead to negative self-evaluations and contribute to the feelings of inadequacy or depression. Indeed, neuroimaging studies have found altered functioning in emotional regulation circuits, including reduced connectivity between the PFC and amygdala in individuals with problematic social media use. Another crucial aspect to consider is the impact of social media on empathy and social cognition. The ability to understand and share the feelings of others relies on a network of brain regions known as the empathy network, which includes the anterior insular, anterior cingulate cortex, and parts of the prefrontal cortex. Interestingly, studies have shown that reading emotional content online activates similar brain regions as witnessing emotions in persons. However, the lack of nonverbal cues in digital communication may be altering how we process social information. For instance, a 2019 study using functional near-infrared spectroscopy, short FNIRS, found reduced activation in the right temporoparietal junction, short TPJ, a region crucial for theory of mind and perspective taking during online compared to face-to-face interactions. This suggests that digital interactions may not engage our social cognitive networks as fully as real-world interactions, potentially impacting our ability to accurately interpret others' emotions and intentions online. Furthermore, the anonymity and physical distance inherent in many online interactions may be affecting activity in brain regions involved in impulse control and moral decision-making, such as the ventromedial prefrontal cortex, VMPFC for short. This could partially explain phenomena like online disinhibition and cyberbullying.
Neuroplasticity and cognitive enhancement, harnessing the positive potential. While much of our discussion has focused on the potential negative impacts of social media on the brain, it's crucial to recognize that not all neuroplastic changes induced by digital technology are detrimental. In fact, when used mindfully, social media and other digital tools have the potential to enhance cognitive function and promote positive neuroplasticity. Let's explore some ways in which digital interactions can potentially benefit brain function. First, cognitive flexibility. The rapid task switching often required during social media use may, in moderation, enhance cognitive flexibility. This involves regions like the frontoparietal control network, including the dorsolateral prefrontal cortex and posterior parietal cortex. Second, information processing. Exposure to diverse content on social media can stimulate the brain's information processing capabilities. This engaged regions like the inferior frontal gyrus and the anterior insula, which are involved in integrating and evaluating new information. Third, social cognition. Despite the limitations of online interactions, they can still provide opportunities to practice social skills, in theory of mind potentially strengthening neural networks involved in social cognition, such as the medial prefrontal cortex and temporoparietal junction. Fourth, creativity. Platforms that encourage content creation like TikTok or Instagram may stimulate creative thinking. This engaged the default mode network and its interactions with the executive control network, potentially enhancing divergent thinking abilities. Fifth, learning and memory. Educational content on social media can promote learning, potentially enhancing synaptic plasticity in the hippocampus and its connections with cortical regions involved in long-term memory storage. To harness these potential benefits while mitigating risks, it's crucial to approach social media use mindfully. This involves being intentional about the content we consume and how we interact online. Research suggests that active social media use may be more beneficial than passive use. Active use engages higher-order cognitive processes and may promote more positive neuroplastic changes. Moreover, using social media as a tool for learning and skill development can be particularly beneficial. For instance, following accounts that share educational content and participating in online communities focused on skill sharing can stimulate neural pathways involved in learning and memory. It's also worth noting that the brain's capacity for neuroplasticity means that we can actively shape how social media affects us. By practicing mindfulness and metacognition during social media use, we can strengthen neural circuits involved in self-regulation and cognitive control. Techniques like mediation or cognitive behavioral strategies can enhance activity in prefrontal regions, potentially counteracting some of the less desirable neuroplastic changes associated with excessive or problematic social media use. In conclusion, while it's important to be aware of the potential negative impacts of social media on brain function, we shouldn't overlook its capacity to promote positive neuroplasticity and cognitive enhancement. The key lies in using these digital tools mindfully and intentionally, leveraging their potential to simulate learning, creativity, and social connections while maintaining a balanced approach to technology use. Conclusion and future directions. As we've explored throughout this video, the relationship between social media and the brain is complex and multifaceted. From the molecular mechanisms of word processing to large-scale changes in brain structure and function, social media is reshaping our neural landscape in profound ways. We've seen now how these digital platforms engage our dopaminergic reward system, challenge our attention networks, alter emotional processing circuits, and potentially induce both positive and negative forms of neuroplasticity. This intricate interplay between technology and neurobiology raises important questions about the future of human cognition in the digital age. Moreover, as we move towards more immersive digital experience like virtual and augmented reality, understanding the neurobiological impacts of these technologies becomes even more crucial. How will these new forms of digital interactions affect our neural circuits for spatial navigation, embodiment, and social cognition? In conclusion, the neuroscience of social media represent a fascinating frontier in our understanding of brain-environment interactions. By continuing to investigate these complex relationships, we can work towards creating digital ecosystems that support rather than subvert our cognitive and emotional well-being. As users of these technologies, understanding their neurobiological impacts empowers us to make informed decisions about our digital habits. And as a society, this knowledge can guide us in shaping policies and designing technologies that respect the intricacies of the human brain. Thank you for joining us on this exploration of the neuroscience of social media. As we continue to navigate the digital age, let's strive to use technology in ways that enhance rather than hinder our remarkable neural capacities.